Hi, I'm Anna, and my project was investigating candidate enzymes for resin glycoside biosynthesis in the morning glory family. I know that's a mouthful, but it'll make sense, so just stick with me. So just a little bit about me. I'm a student at the University of Florida, and I love studying plants and specifically ecology. And so that's kind of what I work on over there. And this summer, I was in the Mogi lab, and there, oh, sorry. Spoiler. They're up there. Right. So what are resin glycosides? So they are a special class of acyl sugars that are only found in the Convolucid family, which is morning glories. And their structure is really, really important to understanding what I'm doing here. So their defining components are a core made of multiple sugars, one or more hydroxyacyl chains, which you can see are in red, and decorating aliphatic chains or acyl groups. Those are in blue. And so they are created through a process called promiscuous enzyme activity. And so these are actually secondary metabolites. So basically what that means is here you have your regular enzyme activity, right? It's specific to the substrate that it works with most of the time. And so it creates the product that is most commonly seen. But sometimes, especially in the absence of that primary substrate, this enzyme can bind to other similar substrates. And so then you can get a secondary metabolite. And these aren't necessary to function, especially since they only form under certain conditions, but they can provide them a lot of advantageous traits that scientists love to research, some of which are inhibiting weed growth, protecting against pathogens and nematodes, and protecting against herbivory from plants. And a lot of plant tissues that are high in resin glycosides are used in traditional medicine around the world. So obviously, with such a broad range of activity, scientists have been able to characterize them structurally and functionally because they're important molecules. But we still don't really understand fully how they form. So there's two enzyme families that we believe are critical, and those are UDP glycosyltransferases, just UGTs, and BAHD acyltransferases. So here's your resin glycoside, and these are all of the enzymes that we currently think are involved in this process. And so basically these, oh my God, I suck this. So basically these UDP glycosyltransferases are working on creating that sugar backbone and attaching to the hydroxyacyl chain. And these VAHDs are attaching these acyl chains onto resin glycoside. More on that in a second. So I was working on both of these enzyme families simultaneously this summer. So with the UGTs, I was working on finding candidate enzymes to be able to test later on using phylogenomic analysis. And for VAHDs, I was actually helping produce these for testing. So starting with the UGTs, this is UGT activity. So basically what you have is you have a substrate and you have a UDP sugar, and you get a UDP sugar group and you attach it onto a other substrate molecule. And so what we wanted to see is, in Ipomia tricolor, which is morning glory, there were 17 UGT sequences that had been found by previous experimentation in the lab, but we didn't really know what they do. So what we did was we got a large database of other plant UGTs just across the spectrum, and we got their amino acid FASTA sequences, so just the sequence that corresponds to this enzyme, and we aligned those sequences with our candidate sequences from Ipomia tricolor. And then in order to know what they did, we tracked their enzymatic activity of the known plant sequences, and we categorized their activity based on the substrates that they use. So what you see here, these are class of substrates. These are all phytochemicals that act as substrates for UDP glycosyltransferase activity. And they're color-coded more or less based on how related these are to each other, but that's not a hard and fast rule, so don't get too caught up on it. And so now with this, we constructed a phylogenetic tree. So this is our tree, and those are the Ipomia tricolor candidate enzymes. So what we did was we looked at the clades that contain the candidates to figure out more or less what they would be doing. So we were looking for clades that have substrates that can be used for acylating sugar activity, and for clades that are more homogeneous, because the more consistency you have in the activity of your clade, the better chance you have that you're actually going to have the activity you're looking for. So with that, we got this clade. Yellow is terpenes, so it's a group of terpenes that are candidaterin, and this one that has flavonoids. And so that gives us these 4 candidate enzymes. However, there was a lack of consistency among all of these enzymes that we saw on the clades. So if you look here, down at the bottom there, that's what we call a rainbow clade. So you see we have a whole bunch of different colors, and they're not all related to each other very much at all. And so that made it really hard to get some good candidates from this. And these UGTs have a really broad array of what they actually do. So they might not be performing the same activity within the substrate class that they're in, as well as they can use many different substrates. So basically these candidates might not be performing the way that we expect them to. So now these UGTs are going to be further tested to see what their activity actually is. And successful candidates have the right activity can be used to reconstruct this resin glycoside pathway. And so now kind of what that step would be, we're going to show in BAHDs. So this is your BAHD activity. Basically, you have an acyl group donor, that's our 2-methylbutyl-CoA here, and it's attaching it onto the resin glycoside core. And so in order to test this activity, we need 3 things. We need an acyl donor, a BAHD, and also the actual resin glycoside backbone. So that's what I'm going to walk you through here right now, starting with acyl donors. So this is one of the acyl donors that we wanted to test, AIC5-CoA. I'm also going to be referring to it as 2-methylbutyl-CoA. And this is an acyl-CoA. So if you know acetyl-CoA, that's an acyl-CoA. This is a modified version of that, not commercially available, so we had to make it. And in order to make that product, we had to first make the enzyme that makes that product. So we started with 3 mutant plasmids, and we are going to make a triple mutant as well. This was all based on prior research. And so we first made the plasmids with the mutation, put those mutations in E. coli so that they created a lot of these plasmids, put those plasmids into more E. coli that actually could express these enzymes, and then we purified those. And then you can see here that these gels indicate the presence of the protein. And so you can see they're right around the expected size that we were looking for. And so these were used in an enzyme assay to actually acquire the 2-methylbutyl-CoA. So this is the liquid chromatography mass spectrometry result that we have. So you can see at the top, this is our control for normal acetyl-CoA unmodified, and it's present both in one of the mutants as well as the wild type. So we know that this works. But down here in 2-methylbutyl-CoA, which is our mutant, we can see that it's only present in this one and on the wild type. Now, what does that mean? It means we actually did need to make a triple mutant, which was great because that was really a struggle. So now we have our acyl donor. Now we're moving on to BAHDs. So a lot of this was already done before I came, so I really focused on expressing and purifying these enzymes. So these were the 3 that were tested while I was here. So, you know, I know the gel looks a little wonky, but I promise, for the sake of this, it works, okay? Anyway, so we have our BAHDs, and now the last thing we need is our resin glycoside like base structure. And so we can get this with alkaline hydrolysis because this actually isn't available commercially either. So my coworker, Ling Hua, he performed this. And so basically you have your resin glycoside trichlorine A. And so these are the acyl groups that are on it. And so we basically just took them off. So we're only left with the core because we want to see if the BAHEs will actually add them back on. So now that we have our 3 components, we can actually get to the assay, which was done by my mentor, Mohammed Irfan. Anyways, so we did see activity. We saw that one of the candidates, BAHD5, acylated butyryl-CoA to make a resin glycoside. And so basically this is AIC4-CoA, butyryl-CoA. So you're getting the acyl chain and just attaching that onto the trichloric acid. And so this was a little unexpected because number one, it's the only one that actually reacted to any acyl donors because there was multiple tested and nothing reacted to 2-methylbutyryl-CoA, which is supposed to be the best candidate based on prior studies. So, that was interesting. And so from now on, clearly based on the prior results, we don't really know the specific proteins or the specific donors that are actually involved in this process. So we're basically going to be synthesizing a whole lot more of them and testing them out in assays. And then hopefully successful candidates can also be used for this RG pathway reconstruction. And so that was a lot, I know. And scientists think so too. So a lot of scientists are researching resin glycosides because their diversity really makes them great candidates to be used as components for drugs and agriculture research. And they're also really good for people studying evolutionary ecology and to understand the formation of other important compounds that might be similar, especially products imbibing glucosylation, things like that. But they're all currently kind of at a bottleneck. So this research is coming really slow. And so why is that? It's like stuck. Okay, sorry. So this is because they're really difficult to isolate and purify. So to kind of take a look at that, the lab is working on consolidating the databases that contain all of these enzymes that are necessary to figure out their activity as well as their activities. So basically just consolidating all this enzyme activity into one place so that it's more easily available for people to be researching, as well as actually trying to understand this pathway so that it can be better understood so that it can be reproduced and modified for research purposes. So with that, I would like to thank my PI, Gaurav Mogi, my mentor, Mohammed Irfan, as well as everyone else in the lab, Delaney and Megan, of course. And thank you all. So.
Time for maybe one question.
That was a great presentation. Thank you very much. Did you have any experience doing this type of work before you came here, or did you have to all learn it on the fly? Whatnot?
Yeah, none. So I work with like plants on, the actual plant level back where I'm from. So I study like physiology as well as just general plant science. Never took orgo, never took microbiology, never took basically anything in this. I'd only been in lab literally one time for Chem 2, not even for Chem 1 because of COVID. So definitely a learning curve. Learned a lot, made a lot of mistakes, as you can tell by my job. But yeah, I think I got a pretty good hang of it though. So it was really nice.
You did a great job. Thank you.
Thank you.